In this chapter so far, we have been looking at adaptive immunity, which is the third and final line of defense. And we looked at the four different stages of how this plays out, which is the development of lymphocyte diversity, the presentation of antigens, the T cell response, and the B cell response. Now, the final section here, we're going to look at adaptive immunity and vaccination. So adaptive immunity in humans as well as in other mammals is categorized using two different sets of criteria. And when they're combined, you end up with four specific descriptors of the immune state. So firstly, immunity can be either natural or artificial. Natural immunity is any immunity that is acquired during the normal biological experiences of an individual rather than through medical intervention. Artificial immunity is protection from infection that's obtained through medical procedures. This kind of immunity is induced by vaccines or immune serum. Then you can also describe immunity or separate it into either active or passive. So active immunity occurs when an individual receives an immune stimulus, so an antigen, that activates the B and T cells, causing their own body to produce immune substances such as antibodies. Active immunity is marked by several characteristics. One, it creates a memory that renders the person ready for quick action upon re-exposure to the same antigen. Two, it requires several days to develop. And three, it lasts for a relatively long time, sometimes even for life. Now, active immunity can be stimulated by natural or artificial means. Passive immunity, this occurs when an individual receives immune substances, which are usually antibodies, that were produced actively in the body of another human or an animal donor. The recipient is protected for a short time, even though they have not had prior exposure to the antigen. This is characterized by the lack of memory for the original antigen, a lack of protection of new antibodies against that disease, immediate onset of protection, and short-term effectiveness because antibodies have a limited period of function and ultimately the recipient's body disposes of them. Just like active immunity, passive immunity can also be either natural or artificial in origin. So here you see natural immunity, active and passive, artificial immunity, active and passive. So in this case here, what's going on? So after recovering from an infectious disease, a person is generally actively resistant to reinfection for a period that varies according to the disease. In the case of childhood viral infections like mumps, measles, rubella, this natural active stimulus provides nearly lifelong immunity. Other diseases result in a less extended immunity of a few months to years, like pneumococcal pneumonia and shigellosis, and reinfection is possible. Even a subclinical infection can stimulate natural active immunity. So this probably accounts for the fact that some people are immune to an infectious agent without ever having been noticeably infected with or vaccinated for it. Now here, in the second case, we're looking at passive immunity, passive natural immunity. So natural passively acquired immunity occurs only as a result of the prenatal and postnatal mother-child relationship. During fetal life, IgG antibodies circulating in the maternal bloodstream are small enough to pass across the placenta. This natural mechanism provides an infant with a mixture of many maternal antibodies that can protect it for the first few critical months outside the womb, while its own immune system is gradually developing active immunity. Depending on the microbe, passive protection lasts anywhere from a few months to a year. Another source of natural passive immunity comes to the baby by way of the mother's milk. Although the human infant acquires 99% of natural passive immunity in utero and only about 1% through nursing, the milk-borne antibodies provide a special kind of intestinal protection that is not available from transplacental antibodies. Now, moving on to the third one, here we are looking at artificial immunity that is active. So this is produced purposefully through medical procedures, whereas the natural immunity here was acquired through normal life experiences of a human and has nothing to do with medical means. So in this active artificial immunity, vaccination exposes a person to a specially prepared microbial or immunogenic stimulus, which then triggers the immune system to produce antibodies and lymphocytes to protect the person upon future exposure to that microbe. As with natural active immunity, the degree and length of protection vary. Vaccines are also being developed for threats to human health that do not involve microbes at all, such as prostate cancer and heroin addiction. Here, in the fourth case, we see passive immunotherapy, which involves a preparation that contains specific antibodies against a particular infectious agent. Think of the monoclonal antibody treatment that was given during COVID to people who were immunosuppressed or in other ways weak. So pooled human serum from donor blood, gamma globulin, and immune serum globulins containing high quantities of antibodies are what is frequently used. COVID-19 immunotherapy, just a brief touch on what was going on then. In the spring of 2020, in the early days, people became more aware of the option of passive immunotherapy. So this is the convalescent serum, monoclonal antibodies from people who were ill with it and had recovered. Hence, they were convalescent. And this approach is useful when there's no vaccine available or also when someone cannot be vaccinated. Let's look at the history of immunization. So the basic notion of immunization has existed for thousands of years. Probably it stemmed from the observation that persons who had recovered from certain communicable diseases rarely got a second case. The earliest crude attempts involved bringing a susceptible person into contact with a diseased person or animal. The first recorded attempt at immunization occurred in 6th century China. It consisted of drying and grinding up smallpox scabs and blowing them with a straw into the nostrils of vulnerable family members. By the 10th century, this practice had changed to the deliberate inoculation of dried pus from the smallpox pustules of one patient into the arm of a healthy person, a technique that was later called variolation. So variola is the name for the smallpox virus, and that's where the name came from. This method was used in parts of Far East for centuries before it was brought to England in 1721 by an English aristocratic woman named Lady Mary Wortley of Montagu. She had spent 2 years in Turkey with her diplomat husband and had witnessed women healers scratching smallpox material into healthy children's skin. This resulted in a mild case of smallpox, though it did occasionally lead to death. The outcome vividly demonstrates a cardinal rule for a workable vaccine. It must contain an antigen that will provide protection but not cause the disease. So eventually this human, wrong direction. Here we go. Eventually this human experimentation paved the way for the first really effective vaccine, which was developed by the English physician Edward Jenner in 1796. Jenner's work was what gave rise to the word vaccine and vaccination, which now apply to any immunity obtained by inoculation with selected antigens. Jenner himself was inspired by the case of a dairymaid who had been infected by a pustular infection called cowpox. This related virus afflicts cattle, but causes a milder condition in humans. The little girl explained that she and other milkmaids had remained free of smallpox. And to test the effectiveness of this new vaccine, Jenner prepared material from human cowpox lesions and inoculated a young boy. When challenged 2 months later with an injection of crusts from a smallpox patient, the boy proved immune. Definitely a method of testing that would be an absolute no-no today. Now, Jenner's discovery that a less pathogenic agent could confer protection against a more pathogenic one is especially remarkable in view of the fact that microscopy was still in its infancy and the nature of viruses was unknown. At first, the use of the vaccine was regarded with fear and skepticism. When his method proved successful and word of its significance spread, it was eventually adopted in many other countries. In 1980, the World Health Organization declared that smallpox had been eradicated. Now, the only natural forms of passive immunization occur as a fetus develops and encounters selected antibodies that are able to cross the placental barrier, and a newborn nurses and receives IgA in breast milk that is secreted at birth and for a short time afterward. The first attempts at artificial passive immunization involved the transfusion of horse serum. The horse serum contained antitoxins to prevent tetanus and to treat patients exposed to diphtheria. Since then, antisera from animals have been replaced with products of human origin that function with various degrees of specificity. Intravenous immunoglobulin, sometimes called gamma globulin, contains immunoglobulin extracted from the pooled blood of human donors. The method of processing IVIG, this is the intravenous immunoglobulin, concentrates the antibodies to increase potency and eliminates potential pathogens, for example, hepatitis B and HIV. It's most useful in patients who have a diminished ability to mount their own immune response, or if the disease is so fast-acting that it could be fatal before the victim develops their own antibodies, for example, tetanus and rabies. A preparation called specific immunoglobulin is derived from a more defined group of donors. Companies that prepare the specific immunoglobulin obtain serum from patients who are convalescing and in a hyperimmune state after such infections as pertussis, tetanus, chickenpox, and hepatitis B. These globulins are preferable to the intravenous immunoglobulin, the IVIG, because they contain higher titers of specific antibodies obtained from a small pool of patients. Although donated immunities only last a relatively short time, they act immediately and they can help patients for whom no other medication or vaccine proves to be useful. This was demonstrated when COVID-19 patients were treated with SIG, the convalescent sera, before there was a vaccine. Now, looking at artificial active immunity under vaccination, going back to the basic principle behind vaccination, which is to stimulate a primary response and a memory response that primes the immune system for future exposure to a virulent pathogen. If the actual pathogen enters the body, the immune response will be immediate, powerful, and sustained. Vaccines have profoundly reduced the prevalence and impact of many infectious diseases that were once common and often deadly. Initially, the emphasis was on immunizing babies and children against formerly common childhood diseases like measles, mumps, and rubella. Recent years, we've seen an additional push to immunize adolescents and adults against agents such as HPV, human papillomavirus, Streptococcus pneumoniae, and herpes zoster, which causes shingles. Vaccines are also being developed for threats to human health that do not involve microbes at all. Now we're going to look at how vaccines are prepared and how they are made as safely as possible. Because there was so much unfounded skepticism about vaccines, please take the time to read the purple, orange, and green infographics about how vaccines are developed and tested. We're going to look at those. You'll see that the process actually takes years. And during COVID-19 pandemic, vaccines were released after an emergency use authorization. You might remember this. This was a provision where vaccines that are in phase 3 can be approved provisionally. So they haven't finished that phase, but they're still released. But massive information about safety has to still be included in the application. So because of the health emergency that COVID-19 posed, the government provided resources for the development and testing to occur in record time. Safety and quality were not compromised in this. Now, the qualities of an ideal vaccine. So, innatural immunity in infectious agents stimulates a relatively long-term protective response, whereas in artificial active immunity, the object is to obtain the same response with a modified version of the microbe or its components, definitely not to get sick. So, qualities of an ideal vaccine are: it should protect against exposure to natural wild forms of the pathogen. It should have a low level of adverse side effects or toxicity and not cause harm. It should stimulate both antibody, B cell response, as well as cell-mediated T cell response. It should have long-term lasting effects. It should produce memory. It should not require numerous doses or boosters, and it should be inexpensive, have a relatively long shelf life, and be easy to administer. Vaccines can be broadly categorized as either whole organism or part of organism preparations. Under these, you have subcategories. So the whole cells or viruses, you could have either killed cells or inactivated viruses, or live attenuated microbial cells or viruses. Under the vaccines that deliver on an antigenic component of the microbe in question, you can have 4 different kinds. You can have microbial surface structures that are either isolated from cultures of cells or viruses, or structures that are produced using recombinant DNA technology. The second kind would be vaccines containing DNA or mRNA that code for microbial surface components. These nucleic acids are enclosed in a lipid vesicle. Third kind would be surface subunits conjugated with proteins, often from other microbes, to make them more immunogenic, called conjugated vaccines. And finally, unrelated viruses. These are called viral vectors, and they carry RNA or DNA for a surface component of a microbe. Here we're looking at types of viruses. You see on the top the killed cell or the inactivated virus, maybe by heat, maybe by chemicals, and then it can be used to vaccinate. And in the lower one, you see the live attenuated, so weakened. Attenuated means it's weakened. Its virulence is eliminated or reduced. So it's still alive, has the same antigenicity. And here as well, you have the same antigenicity, and then you're making vaccines out of that. So an example here would be the flu virus. Here you would see measles, mumps, the MMR, measles, mumps, rubella vaccine. The other kind that we're looking at are the antigenic component vaccines. Here you see a surface antigen that has been used, example Hep B. Here you see the mRNA inside a lipid vesicle, that would be your COVID-19. Here you see surface antigens complexed with protein to increase antigenicity. So it's similar to this, but with another step added on. Haemophilus B, Haemophilus influenza type B conjugated vaccine. And then here you see third-party harmless virus with DNA or mRNA for surface protein of desired virus being injected. So that would be the COVID-19 J&J, the Johnson & Johnson vaccine. mRNA vaccine technology. Now, this is something where you are seeing that actually a part of the mRNA, of the sequence of the cell, has been taken out and used as a vaccine. So this is what you're seeing here, with the mRNA inside the lipid vesicle, injected into a human being. Inside the cell, in the nucleus, now you've got the mRNA, and you end up with multiple spikes being produced. These, because of that code being for the spike protein. So here you had the RNA coding for spike protein. That's what got injected. So you'll have those spikes produced. Because of the spikes being produced on these human cells, you end up with T and B cells becoming sensitive to it. And now you have this protection within your body. When an actual virus comes into the cell, you will see them going and attaching, and they'll be able to neutralize it. How are vaccines administered? So most of them are administered subcutaneous, intramuscular, or intradermal. One kind of the influenza vaccine comes in the form of a nasal spray. Oral or nasal vaccines are available for only a few diseases, but they have some distinct advantages. An oral or nasal dose can stimulate protection, IgA, on the mucous membrane of the portal of entry. So oral and nasal vaccines are also easier to give than injections and are more readily accepted for obvious reasons. Now, some vaccines require the addition of a special binding substance or an adjuvant. The adjuvant is a compound that enhances immunogenicity. It prolongs antigen retention at the injection site. This adjuvant precipitates the antigen and holds it in the tissues so that it will be released gradually. Its gradual release presumably facilitates contact with antigen-presenting cells and lymphocytes. This helps involve the innate immune system as well. Booster shots. Now, everybody has definitely heard of a tetanus booster shot. So the ideal situation, which is only sometimes achieved, is when a vaccine provides complete protection from a particular disease for a lifetime. But after a vaccine has been given to a large number of people, scientists can determine whether the vaccine needs to be boosted, which means another dose needs to be administered to shore up its long-term effectiveness. So we know that tetanus vaccines should be boosted every 10 years or so. The measles vaccine given in infancy calls for a booster at age 5 or 6. During the COVID-19 pandemic, we learned that the original vaccines should be followed by booster vaccines for maximum immunity. This raised an ethical question for wealthy countries when there was a shortage of vaccines in less wealthy countries. Should the precious vaccine doses be used on those who already were fully vaccinated, or made available to countries where a very low percentage of people had achieved the first vaccine regimen? Question worth answering, yeah? Now looking at vaccines for non-infectious conditions. The vaccination strategies are now under intense investigation for prevention and treatment of non-infectious diseases. One of the best examples is vaccination for Alzheimer's disease. Researchers are examining whether administering a peptide that is found in the brain plaques of Alzheimer's patients as a vaccine can lead to prevention of the disease. The principle is the same as with vaccines against microbial infections. If T and B memory cells that recognize these disease-inducing proteins can be created in a host via vaccination, once the proteins begin to form in the patient, the memory response eliminates them before they cause damage. One of these vaccines is in human trials now. Immunotherapy as a treatment for cancer has been in use now for some years. In these situations, T cells or dendritic cells are removed from the patient. In the laboratory, the cells are sensitized to known cancer antigens. These activated cells are then injected back into the patients in the hope that the actual tumor antigens match the ones used for activation in the lab, and the patient's own immune system will attack the tumor. Side effects of vaccines. So all vaccines carry the risk of side effects. The vast majority of side effects are mild. Soreness at the infection site or injection site, injection site, and possible short-term flu-like symptoms, because so many of these symptoms are caused by the body's immune response, such as release of interferons, rather than the vaccine itself. Serious side effects are very rare. The most common serious side effect is the risk of anaphylactic shock. COVID vaccines were seen to result in anaphylaxis in about 5 cases out of 1 million. This risk is the reason that you were asked to stay on the vaccination site for 15 minutes before leaving. On the other hand, in its first year, COVID vaccination is thought to have saved approximately 20 million lives worldwide. Scientists also estimate that 1/3 of the over 1 million US deaths could have been prevented by vaccination. Some people have attempted to link childhood vaccinations to later development of diabetes, asthma, and most prominently autism. However, the original 1998 scientific paper that suggested the vaccines might be responsible for autism was entirely discredited, and the principal author's medical license was revoked after authorities found the research and its claims to be fraudulent. In 2019, the World Health Organization named vaccine hesitancy as one of the top 10 threats to global health. And this was before the COVID-19 pandemic. The biggest cause of vaccine hesitancy seems to be vaccine misinformation. Scientists have recently conducted a study with over 17,000 participants, almost half of them identifying as healthcare students or healthcare workers. They found an alarming amount of misconceptions even among this population. For example, in the non-healthcare population, only 29% of correspondents correctly agreed with this statement: you cannot get the flu from the flu vaccine. Among healthcare students or workers, only 49% agreed with that statement. In another example, 36% of all participant studies agreed or were unsure of the statement: there is evidence that the mercury in the measles vaccine can cause autism. In fact, the measles vaccine has never contained mercury and has been shown beyond a doubt not to cause autism. After the first COVID-19 vaccines were approved, the amount of vaccine misinformation grew exponentially. As mentioned earlier, experience with now billions of administered doses has shown that it is one of the safest vaccines on the market. So, vaccine development, approval, and monitoring. So, looking at how a new vaccine is developed, approved, and monitored. Before a new vaccine is ever given to people, extensive lab testing is done, and this can take several years. Once the testing in people begins, it can take several more before the clinical studies are complete and that the vaccine is finally licensed. The FDA sets rules for the 3 phases of clinical trials to ensure the safety of volunteers, and they test the vaccines with adults first. So phase 1, 20 to 100 healthy volunteers. Is it safe? Does it work? Serious side effects? How's the size of the dose related to side effects? Phase 2, several hundred volunteers. Most common short-term side effects. How are their immune systems responding? And phase 3, hundreds of thousands of volunteers. How do people who get the vaccine and people who do not get the vaccine compare? Is it safe? Is it effective? What are the most common side effects? FDA licenses it only if it's safe and effective and if the benefits outweigh the risks. Vaccines are made in batches called lots. Manufacturers must test all lots to make sure they're safe, pure, and potent, and each lot can be released only once FDA approves. And FDA, in fact, inspects manufacturing facilities regularly to ensure quality and safety. Now, looking at how a vaccine is added to the US recommended schedule, you have an advisory committee on immunization practices, and this is a group of medical and public health experts. Members of the American Academy of Pediatrics and the American Academy of Family Physicians add their advice. All available data are examined before a recommendation is made. Questions asked, like how safe is it when given at specific ages? How well does it work? How serious is the disease? How many children would get the disease the vaccination prevents if we didn't have the vaccine? So until the CDC director reviews and approves the ACIP recommendations, they're not official. And then a new vaccine to protect against a disease is added to the immunization schedule. How is the safety monitored moving forward? So FDA and CDC closely monitor vaccine safety now that hundreds of thousands of people are receiving it, looking for rare events such a large group would reveal. The purpose of this monitoring is to look for adverse effects or side effects, and monitoring after its license helps make sure that if there are any possible risks associated with it, they would be identified. The vaccine adverse effect reporting system involves collecting and analyzing reports of any adverse events happening after vaccination. Anyone can submit this: a parent, a patient, a healthcare professional. And then there is the vaccine safety data link. You have a network of all the healthcare organizations across the US, healthcare information for a population of over 9 million people, and they're using this vaccine safety data link to conduct studies to evaluate the safety of vaccines and determine if side effects are actually associated with vaccines or not. And with that, we end this chapter. I do want to add a tiny bit more information, even though there is no slide actually for this. There are still dozens of bacterial, viral, protozoal, and fungal diseases that remain without a functional vaccine. So right now, we don't have a reliable vaccine for HIV or AIDS, various diarrheal diseases, respiratory diseases, and worm infections. And these infections affect over 200 million people per year worldwide. It's a serious issue. So there's promising technology that is the use of nanoparticles made of surface proteins of the virus of interest. So if these proteins are manufactured inside eukaryotic cells, post-translational modifications can be successfully made, and then the protein ends up looking like it would in nature. So in nanoparticle vaccines, the fully formed surface protein is the vaccine. It doesn't need the human cells it is injected into to do any translating or any modification. A second promising strategy is the use of gene editing techniques, CRISPR, to engineer our own B cells to express the antibodies that we design. COVID-19 has really highlighted a really important need for more and better vaccines, yes, but even more than that, the more equitable distribution of existing vaccines. We see that low and middle income countries struggle to have access to vaccines that the industrialized world takes for granted. So the WHO has created a program called COVAX to ensure international distribution of the vaccine. And this was during COVID that this was created. But this didn't succeed. By the end of 2022, we found that 80% of people in high-income countries were vaccinated at least once, and only 10% of low-income countries were. Solving this problem is part of a much bigger problem of closing the gap between the haves and have-nots on a global scale.